Hello, I'm back to read part 7 of our Rotten Reads and Essay series. This is the last part. Supplement Natural Genome Remodeling from the essay Evolution and the Illusion of Randomness. And this part is in gray, so this is kind of the conclusion here. In her 1983 Nobel address, geneticist Barbara McClintock cited various ways an organism responds to stress by, among other things, altering its own genome. Some sensing mechanism must be present in these instances to alert the cell to imminent danger, she said, adding that a goal of the future would be to determine the extent of knowledge the cell has of itself and how it utilizes this knowledge in a thoughtful manner when challenged. Subsequent research has shown has shown how far-seeing she was. It is now indisputable that genomic change of all sorts is rooted in the remarkable expertise of the organism as a whole. By means of endlessly complex and interweaving processes, the organism sees to the replication of chromosomes and dividing cells, maintains surveillance for all sorts of damage, and repairs or alters damage when it occurs, all with an intricacy and subtlety of well-gauged action that far exceeds at a molecular level what the most skillful surgeon accomplishes at the tissue level. But it's not just a matter of preserving a fixed DNA sequence. In certain human immune system cells, portions of DNA are repeatedly cut and then stitched together in new patterns, yielding the huge variety of proteins required for recognizing an equally huge variety of foreign substances that need to be rendered harmless. Clearly, our bodies have gained the skills for elaborate reworking of their DNA, and we will see further in many different ways. Depending on stage of development, cell type, and state of health, among other things, our cells convert millions of their genomic letters, most often the letter C, standing for cytosine base, to an altered letter in a process known as DNA methylation. A new letter, 5-methylcytosine, is often referred to as the fifth base of the genome, and it has profound implications for gene expression that are far too extensive to survey here. The organism also contrives to affect several other kinds of DNA letter changes. The DNA sequence, it turns out, is subject to intense revision through its participation in life of the larger whole. More emphatically and with remarkable nuance, the organism contextualizes its genome and makes no sense to say that these powers of contextualization are under the control of the genome being contextualized. Thus, the human genome yields itself to a radical and stable redefinition of its meaning in the extremely varied environments of some 250 different cell types and thousands of subtypes found in brain and muscle, liver and skin, blood and retina. It is well to remember that the genes in your stomach lining and the genes in the cornea of your eye are supposed to be the same genes, and yet the immediate context makes very different things out of them. And an especially revealing case of contextualization occurs when the genome fits for the need of all the varied cells of a worm-like larvae, but subsequently pressed into perfectly adequate service for the entirely different cell types and different bodily organizations and different overall functionings of a graceful airborne butterfly. The genome, it appears, is to one extent or another like clay that can be molded in many different ways by the organism as a whole according to contextual needs. Jumping for change. Quite aside from such contextualization, it has long been known that the organism generates altogether new genetic material by duplicating entire genes, modifying them, and supplying them with regulatory elements. This can occur through direct duplication of genes or even larger chromosomal segments, and also through reverse transcription, whereby messenger RNA molecules produced from DNA are transcribed back into new DNA, which can then be modified. But the array of mechanisms underlining the origin of new genes is compelling, extending way beyond the traditional well-studied source of gene duplication, writes Henrik Kesserman of the Center of Integrated Genomics in Switzerland. In a broad overview of the relevant studies, Kesserman documents a dizzying variety of techniques by which the organism diversifies and enlarges its genetic repertoire. For example, two duplicated genes can, via a number of different pathways, fuse into a single chimeric gene. And not only protein coding RNAs, but also small regulatory RNAs can be reverse transcribed into DNA and their functions diversified. And again, various repetitive and mobile elements called transposons, transposons can move around the gene in the genome, often being duplicated in the process and then co-opted either as a new protein coding gene or new regulatory genes. Let's pause for a moment to look a little more closely at these transposons. It is now undeniable, writes a team of researchers from the United States, Canada, Spain, and the United Kingdom. That transposable element, historically dismissed as junk DNA, have had an instrumental role in sculpting the structure and function of our genomes. Directly and indirectly, transposing elements have been found crucial in many aspects of genome organization and renovation. And the diverse means by which the cell employs and regulates them has only begun to be delineated. These transposons, I know that's not right, also known as jumping genes, cute. Who's this? Leaping Larry, who's at lizards, leaping lizards, whose discovery led to Barbara McClintock's Nobel Prize. Well, good for her. May hold the key to a puzzle about inbred mice. Such mice with their perfectly matched genes are sometimes reared in the laboratory under the strictest and most identical conditions possible. The frustration for researchers, according to Fred Gage, a neuroscientist at the Salk Institute for Biological Studies in San Diego, is that you control for everything you can, and in behavioral tests, the variance is enormous. Even within a single litter, one mouse will be usually smart, another below average. Gage and others are proposing that jumping genes help account for this otherwise mysterious diversity. Whatever may be going on with the mice, it has now been shown that transposons move around in the developing mammalian brain, altering the genome from cell to cell. They provide enough diversity among neurons, according to Gage, so that, quote, you can optimize their response to the variety of environments you might encounter throughout life. And now it is being found that transposons also jump in other cell types much more readily than was previously thought. This particularly includes various cells of the early embryo, in which case each genetically altered cell propagates its changes into a subset of the mature organism's tissue, making them genetically distinct from other tissues. Given how often this may happen in the early embryo, there may be much more genomic variation within individuals than most researchers had assumed, write one reporter in Science. None of this looks particularly haphazard. An embryonic stem cell, the regulatory DNA elements known as enhancers of gene expression, contain an elevated number of transposons. The germ cell, of which I will have more to say in a moment, are also especially susceptible to these mutable or mobile elements. The cell type specific and DNA element specific nature of transposon activity points to a meaningfully orchestrated process. In general, there is a bias for many transposable elements to insert themselves upstream of transcription. There's a bias for many transposable elements to insert themselves upstream from transcription start sites, which prevents damage of functional coding elements and enhances the potential for a constructive regulatory change. Are transposon smear parasites? An extraordinarily profound role for jumping genes has just recently come to light with the announcement by Yale University researchers that the evolution of a placental development and hence prolonged pregnancy in mammals was intimately bound up with the regulatory role of transposons. The Yale team found that a network of 1,532 genes recruited for expression in the human uterus, but not in marsupials, a mammalian group whose members give birth to underdeveloped young a mere 2 weeks after conception, is coordinated by transposons. We used to believe that changes only took place through small mutations in our DNA that accumulated over time, remarked the lead researcher in the project, Gunther Wagner. But in this case, we found a huge cut and paste operation that altered wide areas of the genome to create a large-scale morphological change. The study's authors say that their findings strongly support the existence of transposon-mediated gene regulatory innovation at the network level, a mechanism of gene regulation first suggested more than 40 years ago by McClintock. Transposable elements are potent agents of gene regulatory network evolution. It is no wonder then that when genomic researcher David Hassler of the University of California, Santa Cruz, was asked in the journal Cell, what has been most surprising about the human genome? One of the things he cited was mounting evidence that transposons play a critical role in the turnover and reinvention of regulatory elements in DNA. And responding in Science to report about the work on jumping genes in mammalian brains, Southern Illinois University neuroscientist David G. King wrote that the dismissive dictum, mutations are accidents, has grown obsolete, adding the protocols for the spontaneous, non-accidental production of genetic variation are deeply embedded in genomic architecture. One other remark about transposons, they exemplify a growing and, for biologists, embarrassing class of cellular constituents that were initially dismissed as more or less functionless, simply because they didn't fit into any kind of neat, but now hopelessly outmodeled digital coding scheme, linking DNA as master cause to RNA as precisely programmed mediary to protein as definitive final result. Making up a sizable portion of the human genome, transposons are to this day often referred to as junk or parasitic elements, because they play a particular prominent and still barely explored role in the germline. And one often hears about the germ cell's defensive mechanisms to protect itself from these highly mobile, selfish elements with their genome reconstructing potentials. How this kind of thinking could go on for many years without most biologists suspecting a positive role for transposons as a genome remodelers with potentially powerful implications for evolution is a great mystery. Certainly, transposons, like everything else in the cell, are subject to intense oversight by their larger context, and viruses may indeed have played a role in their origin, as many suppose, but this hardly makes them more mere parasites in the organism that have so intently taken them up and put them to use out of thin air. When transposons, the organism reshapes its genome through elaborately organized and synchronized processes, often affecting considerable stretches of DNA. But even more striking, Kessman's notes in the recent discovery of protein coding genes being composed from scratch, that is, from non-protein coding genomic sequence, altogether unrelated to pre-existing genes or transposable sequences. He cites a famous 1977 paper by the preeminent French biologist Francois Jacob to the effect that the probability for creation of a new protein coded genes de novo from scratch by random processes is practically zero. Such creation was widely thought to be virtually impossible, and yet Kesserman goes on, recent work has uncovered a number of new protein coding genes that apparently arose from previously non-coding or non-repetitive DNA sequences. If we take seriously Jacob's practically zero probability for random de novo assembly of functional protein coding genes from non-coding DNA sequences, then given that such assembly does in fact somehow occur, the obvious thing is to suspect is that the process is not random, nor does the scale of the problem as it is now emerging look trivial. There is, we're told by two biologists working in Germany, one at the Max Planck Institute for Evolutionary Biology and one at Christian Albrechts University, accumulating evidence that de novo evolution of genes from non-coding sequences could have an important role in a class of genes representing up to 1/3 of all of the genes in all genomes. The seemingly unabridged gap between practically zero and the recent extraordinary claims invites evolutionary geneticists to do a lot of soul searching. Concerted change in the germline. There is nothing in the picture so far to suggest that when turning our attention to genetic change and reproduction, we will find much evidence of randomness. Everything we've looked at so far occurs in germline cells as well. But in these cells, we witness additional powers of change that could be hardly exceeded. Nowhere, for example, do we see the genome more concertedly reshaped than in the two meiotic cell divisions leading to the formation of gametes in sexual reproduction. Gametes? Gamete? I know that word too. It's been a long time. Anyway. A choreography we hear described in the accompanying main article as in meiotic ballet. Meiotric. I just figured the more I read, the better I'll get at all of these words. One of the central features of this ballet, referred to as chromosomal crossover or genetic recombination, involves an insistent reshuffling of stretches of DNA between chromosomes, resulting in genetic variation in the offspring. You could hardly imagine a more careful and delicately staged dance than the one resulting in chromosomal crossover. And with researchers speaking of recombination hotspots and all sorts of regulation, we can be sure it is not at all random. As usual in the cell, many different factors within the larger whole come to bear on any specific point. As in the case for transcription, no single type of DNA site, transcription factor, or histone modification can account for the regulated positioning of all recombination. Instead, these elements function combatorily with a potential for synergism, antagonism, and redundancy to establish preferential sites of action by mediatoric recombination protein complexes. Context, as always, figures strongly and non-randomly in shaping and redirecting local activities. Kessman further points to studies in animals showing that the testes play a potentially central role in the process of gene birth and evolution. For example, there is an overall propensity of young retrogenes, genes copied back into DNA by reverse transcription from RNA, to be expressed in the testes. The testes may represent a crucible for new gene evolution, allowing novel genes to form and evolve and potentially adopt functions in other somatic tissues with time. Likewise, pluripotent cells, such as stem cells, which bear certain similarities to germline cells, possess genomes that are amazingly plastic. The incredible plasticity of pluripotent genomes is a notable discovery and reveals the view of an unexpectedly dynamic mammalian genome for many of us. Powers of change converging from all sides. In sum, as Kessman writes, recent work in genomics has laid bare an astounding diversity of mechanisms underlying the birth and more recent genes. Almost any imaginable pathway to new gene birth seems to have been documented by now, even those previously deemed highly unlikely or impossible. Thus, new genes have arisen from copies of old ones. Protein and RNA genes were composed from scratch. Protein coding genes metamorphosed into RNA genes. Parasitic genome sequences were domesticated. And finally, all of the resulting components also readily mixed to yield new chimeric genes with unprecedented functions. None of this is yet to mention the way the organism massively restructure, and regulates its genome through the intricate remodeling of chromatin. Is that right? Chromatin, the DNA protein RNA complex compromising our chromosomes. Or the way it shapes the dynamic three-dimensional organization of cell nucleus, which in turn has a great deal to do with how genes get expressed. See the first article in this set, Getting Over the Code Delusion, summer of 2010. Even regarding the bare DNA sequence in the narrowest sense, Italian geneticist Vittorio Sagaramella, after noting the various alterations of the sequence throughout the cells of our bodies, was led to ask, which is our real genome? He adds, the human genome seems more complex but less autonomous than originally believed. Less autonomous because many concerted activities of the organism are brought to bear on it. And there is still much more we could have spoken about. For example, there is a consensus today that entire organelles of the cell originated in evolutionary history through a kind of cooperative fusion of distinct microorganisms, a process requiring an almost unimaginable degree of intricate coordination among previously independent life processes. There is also well-demonstrated reality of lateral gene transfer, which looks like invalidating the image of an evolutionary tree, especially at the level of simpler organisms. Repeated horizontal exchanges of genetic material between distinct species make large portions of the tree look more like a complex web. Then again, there is good evidence that viruses have played a major role in contributing to the genomes of more complex organisms, including mammals and humans. I've been reading a lot about that. If you're interested in, if you're just showing up, I have a lot of things about how the viruses alter our genetic codes in previous articles. In all this, we find organisms bringing their separate, highly coordinated life processes to bear upon each other in a symbiotic and other interactive manner that can be no more described as random than can, say, the complex and elaborately orchestrated mating process we see among sexually reproducing organisms. Then, too, we could have looked at convergent evolution and the way it commonly involves changes to corresponding genes in a widely different organisms, which implies a surprisingly predictability underlining the genetic basis of evolutionary changes. And there is still rapidly rising interest in a kind of neo-Lamarckian, epigenetically mediated inheritance of acquired characteristics. But we've already seen enough to realize that by one means or another, the organism pursues its own genomic alterations with remarkable insistence and subtlety. Where is randomness? All these revelations about coherent genomic changes have prompted University of Chicago's geneticist James A. Shapiro to speak of natural genetic engineering. We have progressed from the constant genome, subject only to random, localized changes at a more or less constant mutation rate, to the fluid genome, subject to episodic, massive, and non-random reorganizations capable of reproducing new functional architectures. Crucially, genetic change is almost always the result of cellular action on the genome. Likewise, two geneticists from the University of Michigan Medical School, writing in Nature Reviews Genetics, remember how it was previously thought that most genomic rearrangements formed randomly. Now, however, emerging data suggests that many are non-random, cell type, cell stage, and locus-specific events. Recent studies have revealed novel cellular mechanisms and environmental cues that influence genomic rearrangements. Think those FCs. Bear in mind that we've been looking at the one aspect of organismal functioning and mutational aspect, where we are assured most confidently that blind chance or randomness becomes visible within the evolutionary process. Certainly from the organism's side, we see nothing to suggest any fundamental role for randomness. The accompanying article explores the question in a larger context, where our understanding of evolutionary fitness becomes crucial. And that's the end of that article. Here's all of the notes that are referenced. You can go look up this article yourself and take a peek. If you find these articles interesting, do the like, subscribe thing and leave me a comment and tell me, would you like me to read some more of these articles? Do you find them as interesting as I do? I saw several things that were referenced in here that sounds interesting enough to read, I think. And so if I'm going to read them for myself, I might as well read them for you. And also find that reading in this way, as if I'm reading a children's book to myself, somehow helps me understand it a little bit better. Thanks for listening to all of my rotten reads and essay parts.